Developmental biology. Basic concepts of development: self-age, potency, determination and differentiation, commitment, specification. Three types: autonomous, conditional, syncytial, genomic equivalence and cytoplasmic determinants, morphogenic gradients, genomic imprinting. A stem cell concept: progenitor cells, adult stem cells, mesenchymal stem cells, multipotent adult stem cells, pluripotent embryonic stem cells, induction, competence, determination and differentiation, morphogenic gradients, cell fate and cell lineages. Then gametogenesis, fertilization and early development, production of gametes, spermatogenesis and oogenesis, ultrastructure of gametes, cell surface molecules in sperm egg recognition in animals like sea urchin and mammals, zygote formation, encounter of sperm and egg, capacitation, acrosome reaction, activation of ovum, amphimixis, prevention of polyspermy, cleavage and blastula formation, gastrulation and formation of germ layers in amphibia. So first, basic concepts of development: cell fate, potency, determination and differentiation. Cell fate. The fate of a cell describes what it will become in the course of normal development. The fate of a particular cell can be discovered by labeling that cell and observing what structure it becomes a part when the fate of cells of our embryo has been discovered. We can build a fate paper, which is a diagram of the organism at an early stage of development that indicate the fate of each cell or region at a later stage of development. And potency, the developmental potential or potency of a cell describes the range of different cell types it can become. That is potency. The zygote and its very early descendants are totipotent. These cells have the potential to develop into a complete organism. Totipotency is common in plants but is uncommon in animals after the 2 to 4 cell stage. As development proceeds, the developmental potential of individual cells decrease until their fate is determined. Then determination. The determination of different cell types, cell fates, involve progressive restrictions in their developmental potentials. When a cell chooses a particular fate, it is said to be determined, although it still looks just like its undetermined neighbors. Determination implies a stable change. The fate of determined cells does not change. Then differentiation. Differentiation follows determination as the cell elaborates a cell-specific development program. Differentiation results in the presence of cell types that have clear-cut identities such as muscle cells, nerve cells, and skin cells. And commitment and specification. Commitment. The determination of a cell to a particular fate can be broken down into two states where the cell can be specified or committed or determined. In the state of being committed or specified, the cell type is not yet determined, and any bias the cell has toward a certain fate can be reversed or transformed to another fate. If a cell is in a determined state, the cell's fate cannot be reversed or transformed. In general, this means that a cell determined to differentiate into a brain cell cannot be transformed into a skin cell. What's a specification? There are 3 general ways a cell can become specified for a particular fate. They are autonomous specification, conditional specification, and syncytial specification. First is about autonomous specification. This type of specification results from cell intrinsic properties. It gives rise to mosaic development. The cell intrinsic properties arise from a cleavage of a cell with asymmetrically expressed maternal cytoplasm determinants, proteins, small regulatory RNAs, and mRNA. Thus, the fate of the cell depends on factors secreted into its cytoplasm during cleavage. Autonomous specification was demonstrated in 1887 by French medical student Laurent Chabry working on tunicate embryos. This asymmetric cell division usually occurs early in embryogenesis. Positive feedback can create asymmetry from homogeneity. In cases where the external or stimuli that would cause asymmetry are very weak or disorganized, through positive feedback, the systems can spontaneously pattern itself. Once the feedback has begun, once again I'm going to read this. This type of specification, which one autonomous specification, results from cell intrinsic properties. It gave rise to mosaic development. The cell intrinsic properties arise from a cleavage of a cell with asymmetrically expressed maternal cytoplasmic determinants, proteins, small regulatory RNAs, and mRNA. Thus, the fate of the cell depends on factors secreted into its cytoplasm during cleavage. Autonomous specification was demonstrated in 1887 by French medical student Laurent Chabry working on tunicate embryos. This asymmetric cell division usually occurs early in embryogenesis. Positive feedback can create asymmetry from homogeneity. In cases where the external or stimuli that would cause asymmetry are very weak or disorganized, through positive feedback, the systems can spontaneously pattern itself. Once the feedback has begun, any small initial signaling is magnified and this produces an effective patterning mechanism. This is normal what occurs in the case of lateral inhibition, in which neighboring cells induce specification via inhibition or inducing signals, notch signaling for example. This kind of positive feedback at the single cell level and tissue level is responsible for symmetry breaking, which is an all or null process where once the symmetry is broken, the cell involved become very different. So symmetry breaking leads to a bistable or multistable system where the cell or cells involved are determined for different cell fates. The determined cells continue on their particular fate even after an initial stimulatory inhibitory signal is gone, giving the cell a memory of the signal. So symmetry breaking leads to a bistable or multistable system where the cell or cells involved are determined for different cell fates. The determined cells continue on their particular fate even after the initial stimulatory inhibitory signal is gone, giving the cell as memory of the signal. The next, conditional specification. In contrast to the autonomous specification, this type of specification is a cell extrinsic process that relies on cues and interactions between cells or from concentration gradients of morphogens. An inductive interaction between neighboring cells is the most common mode of tissue patterning. In this mechanism, one or two cells form a group of cells with the same developmental potential are exposed to a signal or morphogen from outside the group. Only the cells exposed to the signal are induced to follow a different developmental pathway, leaving the rest of the equivalence group unchanged. Another mechanism that determines the cell fate is regional determination, regional specification. As implied by the name, the specification occurs based on where within the embryo the cell is positioned. It is also known as positional value. And this was first observed when mesoderm was taken from the perspective thigh region of a chick embryo, was grafted into the wing region and did not transform to wing tissue but instead into toe tissue. And syncytial specification, this type of specification is a hybrid of autonomous and conditional that occurs in insects. This method involves the action of morphogen gradients within the syncytium. As there are no cell boundaries in the syncytium, these morphogen can influence nuclei in a concentration dependent manner. Then cell lineage. Cell lineage denotes the developmental history of a tissue or organ from the fertilized embryo. This is based on the tracking of an organism's cellular ancestry due to the cell's divisions and relocation as time progresses. This starts with the originator cells and finishing with a mature cell that can no longer divide. This type of lineage can be studied by marking a cell with fluorescent molecules or other traceable markers and following its progeny after cell division. Some organisms, such as C. elegans, have a predetermined pattern of cell progeny, and the adult male will always consist of 1031 cells. This is because cell division in C. elegans is genetically determined and known as utely. UTLY. This causes the cell lineage and cell fate to be highly correlated. Other organisms, such as humans, have variable lineages and somatic cell numbers. Then genomic equivalence and cytoplasmic determinants. Genomic equivalence, the theory that all cells of an organism contain an equivalent complement of genetic information. Genomic equivalence has been confirmed for most cells, but exceptions occur in some animal cells where loss, gain, or rearrangement of nuclear DNA has been observed. Examples of such exceptions include chromatin diminution in somatic cells of some nematodes, selective amplification of rRNA genes in Xenopus oocytes, and DNA excision and rearrangements during mammalian lymphocyte maturation that result in the generation of antibody diversity. Then cytoplasmic determinants are special molecules which play a very important role during oocyte maturation in the female ovary. During the period of time, some regions of the cytoplasm accumulate some of the cytoplasmic determinants whose distribution is thus very heterogenic. They play a major role in the development of embryos' organs. Each type of cell is determined by a particular determinant or group of determinants. All the organs of the future embryo are distributed and operated well thanks to the right position of the cytoplasmic determinants. The action of the determinants on the blastomeres is one of the most important ones. During the segmentation, cytoplasmic determinants are distributed among the blastomeres at different times depending on the species and on the type of determinant. Therefore, the daughter cells resulting from the first divisions are totipotent. They can independently lead to a complete individual. That is not possible after the cytoplasmic determinants have been distributed in the differentiated blastomeres. During the mosaic development, the future embryo contains all the distinct cytoplasmic determinants that are distributed in distinct cells. Regions of the organism differentiate very quickly if each cell contains specific cytoplasmic determinants in the first divisions. Then the cell divides to give all the other cells of its type, and the same process happens in all types of cells in the organism. As a result, in the case of mosaic development, cell totipotence disappears very quickly during segmentation. Indeed, each new created cell determines a new region of the future organism, and it is independent from the other ones. Thus, development is independent from interaction between cells. It is most of all known in certain animals as nematodes, C. elegans, or ascidians, marine animals. Other animals show regulation development, where cells show totipotence for a longer time. In these animals, cytoplasmic determinants are more rapidly distributed between newly created cells. Mammals are an example of animals that undergo regulation development. The zygote already contains the future organization of the embryo through cytoplasmic determinant distribution. So, presumptive territories maps could be established simply by coloring particular organs of the zygote cytoplasm. And morphogenetic or morphogen gradients. A morphogen is a substance whose non-uniform distribution governs the pattern of tissue development. In the process of morphogenesis or pattern formation, one of the core processes of developmental biology is establishing positions of the various specialized cell types within a tissue. More specifically, a morphogen is a signaling molecule that acts differently on cells to produce specific cellular responses depending on its local concentration. Typically, morphogens are produced by source cells and diffuse through surrounding tissues in an embryo during early development, such that concentration gradients are set up. These gradients drive the process of differentiation of unspecialized stem cells into different cell types, ultimately forming all the tissues and organs of the body. The control of morphogenesis is a central element in evolutionary developmental biology. During early development, morphogen gradients result in the differentiation of specific cell types in a distinct spatial order. The morphogen provides spatial information by forming a concentration gradient that subsides a field of cells by inducing or maintaining the expression of different target genes at distinct concentration thresholds. Thus, cells far from the source of the morphogen will receive low levels of morphogen and express only low threshold target genes. In contrast, cells close to the source of morphogen will receive high levels of morphogen and will express both low and high threshold target genes. Distinct cell types emerge as a consequence of the different combinations of target gene expression. In this way, the field of cells is subdivided into different types according to their position relative to the source of the morphogen. This model is assumed to be a general mechanism by which cell type diversity can be generated in embryonic development in animals. Some of the earliest and best studied morphogens are transcription factors that diffuse within early Drosophila melanogaster embryos. However, most morphogens are secreted proteins that signal between cells. A morphogen spreads from a localized source and forms a concentration gradient across a developing tissue. In developmental biology, morphogen is rigorously used to mean a signaling molecule that acts directly on cells, not through serial induction, to produce specific cellular responses that depend on morphogen concentration. This definition concerns the mechanism, not any specific chemical formula. So, simple compounds such as retinoic acid, the active metabolite of retinol or vitamin E, may also act as morphogens. The model is not universally accepted due to specific issues with setting up a gradient in the tissues outlined in the French flag model and subsequent work showing that the morphogen gradient of the Drosophila embryo is more complex than the simple gradient model would indicate. So, proposed mammalian morphogens includes retinoic acid, sonic hedgehog, transforming growth factor beta, TGF beta, bone morphogenic protein BMP, and WNT beta catenin. Morphogens in Drosophila include Decapenda, pelagic, and hedgehog. During development, retinoic acid, a metabolite of vitamin E, is used to stimulate the growth of the posterior end of the organism. Retinoic acid binds to retinoic acid receptors, which acts as transcription factors to regulate the expression of Hox genes. An exposure of embryos to exogenous retinoids, especially in the first trimester, results in birth defects. So, TGF beta family members are involved in dorsoventral patterning and the formation of some organs. So, binding to TGF beta to type 2 TGF beta receptors recruits type 1 receptors, causing the later to be transphosphorylated. The type 1 receptors activate SMAD proteins that in turn act as transcription factors that regulate gene transcription. Sonic hedgehog are morphogens that are essential to early patterning in the developing embryo. SHH binds to the patched receptor, which in the absence of SHS inhibits the smooth end receptor. Activated smooth end in turn causes GLI1, GLI2, and GLI3 to be translocated into the nucleus, where they activate target genes such as PTCH1 and engraved. And next, genomic imprinting. Genomic imprinting was discussed under genetics. So, genomic imprinting in mammalian development, that imprinting might be a feature of mammalian development, was suggested in breeding experiments in mice carrying reciprocal chromosomal translocations. So, nucleus transplantation experiments in mouse zygotes in the early 1980s confirmed that normal development requires the contribution of both the maternal and paternal genomes. The vast majority of mouse embryos derived from parthenogenesis, called parthenogenesis, with two maternal and egg genomes, and androgenesis, called androgenesis, with two paternal or sperm genomes, die at or before the blastocyst implantation stage. In the rare instance that they develop to post-implantation stages, gynogenetic embryos show better embryonic development relative to placental development, while for androgynous, the reverse is true. Nevertheless, for the later, only a few have been described. No naturally occurring cases of parthenogenesis exist in mammals because of the imprinted genes. Then stem cells, induction and competence. Induction organs are complex structures composed of numerous types of tissues. In the vertebrate eye, for example, light is transmitted to the transparent corneal tissue and focused by the lens tissue, the diameter of which is controlled by muscle tissue, eventually impinging on the tissue of the neural retina. The precise arrangement of tissues in this organ cannot be disturbed without impairing its function. Such coordination in the construction of organs is accomplished by one group of cells changing the behavior of an adjacent set of cells, thereby causing them to change their shape, mitotic rate, or fate. This kind of interaction at close range between two or more cells or tissues of different history and properties is called proximate interaction or induction. There are at least two components to every inductive interaction. The first component is the inducer, the tissue that produces a signal or signals that changes the cellular behavior of the other tissue. The second component, the tissue being induced, is the responder. Competence. Not all tissues can respond to the signal being produced by the producer. For instance, if the optic vesicle, presumptive retina, of Xenopus laevis is placed in an ectopic location, in a different place from where it normally forms underneath the head ectoderm, it will induce that ectoderm to form lens tissue. Only the optic vesicle appears to be able to do this, therefore it is an inducer. However, if the optic vesicle is placed beneath ectoderm in the flank or abdomen of the same organism, that ectoderm will not be able to respond. Only the head ectoderm is competent to respond to the signals from the optic vesicle by producing a lens. This ability to respond to a specific inductive signal is called competence. So, competence is not a passive state but an actively acquired condition. For example, in the developing chick and mammalian eye, the PAX6 protein appears to be important in making the ectoderm competent to respond to the inductive signal from the optic vesicle. So, PAX6 expression is seen in the head ectoderm, which can respond to the optic vesicle by forming lenses, and it is not seen in other regions of the surface ectoderm. Then gametogenesis. Gametogenesis is a biological process by which diploid or haploid precursor cells undergo cell division and differentiation to form mature haploid gametes. Depending on the biological life cycle of the organism, gametogenesis occurs by meiotic division of diploid gametocytes into various gametes or by mitosis. For example, plants produce gametes through mitosis and gametophytes. The gametophytes grow from haploid spores after sporic meiosis. The existence of a multicellular haploid phase in the life cycle between meiosis and gametogenesis is also referred to as the alternation of generations. Animals produce gametes directly through meiosis and diploid mother cells in organs called gonads, testes in males and ovaries in females. So, males and females of a species that reproduce sexually have different forms of gametogenesis. First, spermatogenesis in male and oogenesis in female. So, before turning into spermatogonia, the embryonic development of gametes is the same in males and females. Common path gametogonia is usually seen as the initial stage of gametogenesis. However, gametogonia are themselves successions of primordial germ cells. PGCs from the dorsal ectoderm of the yolk sac migrate along the hindgut to the gonadal ridge. They multiply by mitosis, and once they have reached the gonadal ridge in the late embryonic stage, are referred to as gametogonia. Once the germ cells have developed into